as usual, today we will have a discussion. For today's session, we will discuss about cell. Firstly, I would like to ask you, let's pause the presentation and think a little bit about cell. And after you know the definition of the cell itself, you might ask for several questions, such as what is the structure and the function of cells? The second is what are the chemical components of cell? And the last is what process occur in the cell. So without further explanation, let's start to learn about cell. Enjoy! So, as what you see in your screen, this is a picture of cell. And we're gonna discuss about its compartment. So before we learn about cell itself, we have to know the history of discovery and cell theory. The first guy is Robert Hooke in UK. He invented simple microscope shows a cork incision, and then it's continued by Anthony van Leeuwenhoek. He invented a simple microscope has a strange object in a straw choking water. And the third guy is G.B. Lamarck. He stated that every little thing is a collection of cells. And the fourth guy is Henry Dutrochet. He stated that in each cell moves a complex liquid, and the cell is a fundamental part of the organisms. This is the history of the first cell theory discovery. A is the Robert Hooke microscope, which is used to observe thin cork incision. And for picture B, it's the cross-section of the thin section of the cork that Hooke observed using his microscope. As what I mentioned before, the second guy is Anthony van Leeuwenhoek, and this is a microscope Leeuwenhoek picture. This is a simple microscope has a strange object in a straw soaking water. As you can see, this is the picture of the first microscope by Anthony van Leeuwenhoek. Next is cell history and theory. This is statement all life composed of cells, and the theory, cells as units, structural or constituent of body organism. This stated by Matthias Sladen and T. Swan. Based on the number of cells, organisms are divided into two: unicellular, that compose of one cell, and multicellular, which compose of many cells. This is the picture of unicellular, and this is picture of multicellular. This is the more detail of cell history and theory. From Max Schultz, the protoplasm is a structure of basic organism. And then the next is Felix Dujardin. He stated that the most important part of a living cell is a protoplasmic fluid. The third is G. Purkinje. He stated that protoplasm is a living liquid contained in living cells, and the theory is cells are functional units. The next is Rudolf Virchow. Omnicellulae e cellulae, which means each cell comes from the previous cell. And the theory is cells are growth units. And the last is due to technological developments, the cell theory is a unit of heredity. Our first topic about cell is the prokaryotic cell. Prokaryotic cells is a cell which they do not have a nucleus. Its genetic material or DNA is concentrated in an area called a nucleoid, but there is no membrane separating this region from other cell parts. The plasma membranes separate the inner area of the cell from the outside environment. This is the example of prokaryotic cell, which is bacterial cells. And the next is eukaryotic cells. Diverse from the prokaryotic cells, eukaryotic cells have a real nucleus that is enclosed in a nuclear membrane. DNA or genetic material of eukaryotic cells is composed of one or more linear molecules called chromosomes. And all genetic material in eukaryotes stored in the nucleus, and it's separated from the cytoplasm by the nucleus membrane. And the example of eukaryotic cells is animal cell, also plant cell. This is the general cell structure. It has cell membrane, cytoplasm, and then the last is cell organelles. First is cell membrane. Cell membrane is the outer part of the cell, which has some function. They control the exchange of substance as a place of reaction, for example, the light reaction, oxidation, and respiration reaction. The second is as a receptor of excitatory recipient from outside cell protectors. And they also control the substance that enter and exit the cell. And as you can see, composition of cell membrane, there is many kind of protein like alpha helix protein, and then channel protein, peripheral, integral. Also, they compose of cholesterol, glycolipid, carbohydrate, glycoprotein, and then globular protein. And the next picture is the fluid mosaic model of the cell membrane. As what you see before, the composition of cell membrane. Lipid bilayer containing protein, cholesterol, and oligosaccharides. Lipids on cell membranes are called phospholipids. Phospholipid consists of hydrocarbonyl, hydrophobic, which is waterproof, head, which is polar, and it is hydrophilic or love water. The proteins in cell membranes are distinguished into integral protein that is seen penetrating the membrane, peripheral proteins that are on the membrane surface. In cell membranes, there are binding carbohydrates with a protein called glycoprotein or carbohydrates that bind to the head of phospholipids, and it's called glycolipids. The second part of cell is cytoplasm, which is a gel-like material that is located inside the cell membrane and outside the nucleus. So it is between the cell membrane and the nucleus itself. The cytoplasm consists of cytosols and organelles. Inside the cytosol, there is a cytoskeleton that maintains the cell shapes, the mechanical strength, and organizing the cell movement, also chromosome movement in cell division. And this is the clear picture of the nucleus, which consists of genetic material. And the nucleus itself is an organelle that controls all cell activities. As what I mentioned before, the nucleus consists of genetic information in the form of DNA. The nucleus have some important parts. The first is outer membrane and the inner membrane. The core membrane consists of two layers which function as a wrapper as well as a core protector of the cell. In this section, there are pores that allow the exchange of substance with the cytoplasm. And the next is nucleoplasm, which is a gel-shaped core liquid that is rich in ions, proteins, enzymes, nucleotides, and chromatin strands. And then the last is the nucleoli, which compose of a collection of genes that give ribosomal RNA codes. The next part is ribosomes. Ribosome is a small organelle composed of ribosomal RNA, or it's abbreviated as rRNA, and several proteins. Ribosome consists of two subunits that work together in translating mRNA or messenger RNA into the polypeptide chains during the process of protein synthesis. The location of the ribosome, it is free in the cytoplasm and inherent in the endoplasmic reticulum. The endoplasmic reticulum itself consists of two types, which is the smooth endoplasmic reticulum or smooth ER, and then the second is rough endoplasmic reticulum or rough ER. The endoplasmic reticulum itself is a marsupial organelles that play a role in the assembly and processing, as well as the transportation of chemical compounds used inside and outside the cells. Endoplasmic reticulum or ER is related to the two-layer nuclear envelope, or as a channel that connects the nucleus with cytoplasm. As what I mentioned before, it consists of two types, rough and smooth ER. For rough ER, it has ribosomes on its surface. And in contrast, for the smooth ER, they do not have ribosome on its surface. The next part is Golgi Bodies. So Golgi Bodies is apparatus organelles with squiggly membranes that concentrate and wrap proteins to be released into cells. They also play a role in aiding direction or path for the final journey of the protein that has been packaged. This is the analogy of the function of Golgi bodies. It lifts and changes the material inside chemically, produce wax and mucus, for fat transport, for the secretion of protein, carbohydrates, fats, and glycoproteins. It can form lysosome, and it can form digestive enzyme which is not yet active, called zymogen. The next part of organelle is mitochondria. Mitochondria is basically where the respiration takes place aerobic in cells. The mitochondria itself consists of matrix, outer membrane, inner membrane, cristae, F0 and F1 portion. The cristae is used to expand the surface, and the matrix is rich in respiratory enzymes or cytochromes and DNA, RNA, and protein compounds. And then the next part is lysosome. Lysosome is a cell-shaped organelles with somewhat rounded and bounded by a single membrane system. The lysosomes has several functions, such as used to digest the material taken by endocytosis, play a role in the destruction of structures unwanted in a cell. They can release enzyme outside the cell, and for cell self-destruction by freeing all contents lysosome in cells. The next organelles are peroxisomes and glycosysomes. Peroxisomes and glyoxysomes is classified as microbodies. This organelle is enveloped by a single membrane and has the type as what I mentioned before, the peroxisomes and glyoxysomes. The peroxisomes are always associated with other organelles. It also consists of many enzymes, catalase, and oxidase. Catalase can protect cells from destruction by hydrogen peroxide, and the main role are for substrate oxidation, fat metabolism into carbohydrate, and changes in purines in cells. And for the glyoxysomes, it found in plant cells and function to oxidize fatty acids into sugars for plant growth. The next part is chloroplast. Chloroplast is a plastid that contain green, yellow, and red pigments. They have function as organizer of photosynthesis. This organelle has photosynthetic pigments, and the picture is showing you the part of chloroplast itself. The main part of chloroplast are grana and stroma. The grana is encased in thylakoid membranes. And as what I mentioned before, chloroplast consists of photosynthetic pigments, which are the chlorophyll and carotenoids. For photosynthetic pigments, the first one is chlorophyll. Maybe you are familiar with this one. It is a green pigment that mainly absorbs blue, red, purple, and reflects the green light, unless it is covered by other color pigments. And the chlorophyll for photosynthesis were divided into two, which are chlorophyll A and B. And for the carotenoids, the pigments are yellow, orange, red, or brown. It is absorbing the light waves between purple-blue. The other function is protect chlorophyll when the light is too strong, and oxidation by oxygen produced in the photosynthesis process. And after we understand what inside the cell, we will discuss about the process that occur in the cell. However, due to some lacking of time, our discussion about what is occur in the cell to be continued to the next material. So, before end up this session, I would like to give you some exercise. Therefore, you can be more understand about what we have discussed today. As what depicted in the picture, you have to make a table of cells making up the chemical components for each part of the cell itself. The second, please make a mind map about the cell structure and the function of the cell part themselves. And the last part is you have to look at the picture A and B, and you have to mention the organelles in the picture and make a chart of the differences between plant cell and animal cells.